and immunology. And so now we come to our third speaker, Professor Arnaud Marchand, who will look at it from an immunologic point of view and learn us what he learned since the previous webinar. Professor Marchand, please. We don't hear you, Professor Marchand.
Many apologies.
Okay, thank you.
So, thank you, Joris, and thank you for the invitation to speak to this large international audience today. So what I will be talking about are three topics that have been discussed extensively over the last months, and that's the pathogenesis of COVID-19, the role of innate and adaptive immune responses, the possible cross-reactivity between other coronaviruses, non-SARS-CoV-2 coronaviruses, and the role it could play in immunity to COVID-19, and then the immunity induced by SARS-CoV-2 infection itself, and the implications this would have for the coming months for those people who have been infected by the virus. So this is a lot to cover in 15 minutes, so I will touch upon a number of concepts, and I will not have the time to go into the details, but there will be references for the audience to go for if they are interested.
We don't see your slides, sorry.
You don't?
No, I can share them if you want.
Can you see them now?
Yes.
Oh, I thought.
Thank you.
Sorry about that. So this is the overview. Many apologies again. So this is the overview I just mentioned. It was recognized very early on that COVID-19 has, SARS-CoV-2 infection is a spectrum of clinical presentation, and that the critical forms, the severe forms of the disease, are associated with systemic and organ inflammation. We have high levels of cytokines, inflammatory cytokines in the peripheral blood, and we do see infiltration of tissues with inflammatory cells. So when we talked about this in May, this is the working model that we discussed. It was conceived that the immune effectors that are able to control viral infections, including respiratory viruses, macrophages, neutralizing antibodies, and T lymphocytes, that those effector functions do not perform the way they should in the context of severe disease, and what they undertake is what they go for is exacerbated responses. The myeloid cells, especially, but probably also the T lymphocytes, are hyperactivated, produce inflammatory cytokines, and that contribute to tissue damage and dysfunction. At that time, it was proposed that antibodies could have pathogenic role in the context of this disease, and I will come back to that in a few minutes. Since then, a number of studies have been conducted, essentially confirming this working model, but looking more details at the cellular level and molecular level at what's happening primarily in peripheral blood. And those studies have found increased levels of inflammatory cytokines correlating with disease severity, especially IL-6 in intensive care unit patients. These studies, these systems biology studies, have looked in an unbiased way at the total population of immune cells in the blood, looking at the single cell, the transcriptome of those cells, and analyzing diversity of immune cell populations. And among the findings of these studies, we see that type 1 interferon production is reduced in severely infected patients. And so this could contribute to disease severity because, as you know, type 1 interferons are critical for viral control. When we talked in May, we already knew that coronaviruses are actually good at interfering with type 1 interferon responses, preventing type 1 interferon production, and also interfering with type 1 interferon responses, the action of type 1 interferon on target cells. Since then, we've learned that SARS-CoV-2 is doing that very efficiently, and potentially even more efficiently than SARS-CoV-1 and MERS. So the working model we are facing, or we are using now, is that under normal circumstances, when you have type 1 interferon production, that by epithelial cells, for instance, or dendritic cells, this type of interferon can control viral replication. But when this type of interferon production or response is defective, this allows viral replication, and that viral replication will induce monocyte recruitment and will stimulate cytokine production, inflammatory cytokine production, and thereby lead to disease. Two breakthrough studies that have been published recently in Science strongly support a role for type 1 interferon in preventing life-threatening severe COVID-19. In this first study, in-moron errors of type 1 interferon immunity, so genetic defects in type 1 interferon immunity, were detected in a small group of patients, but these are rare mutations in genes coding for proteins involved in type 1 interferon responses. Perhaps more intriguing and interesting is the discovery that about 10% of the patients with life-threatening COVID-19 have auto-antibodies against type 1 interferon. So antibodies that the patients themselves produce and that block the effect of type 1 interferon and thereby their capacity to control the virus. The interpretation of these data is that these patients had these autoantibodies before the infection. So it's not caused by the infection itself, but it was present before and potentially promoted. The production of these antibodies could have been promoted, stimulated by the infection. So producing type 1 interferon, responding to type 1 interferon is critical for protection against severe COVID-19. Another topic that has been discussed in terms of COVID-19 pathogenesis is the potential role of antibodies and their capacity to enhance either infection or disease. As you know, antibodies, when they bind viral particles, they can promote their phagocytosis by macrophages and their destruction. In some viral infections like dengue, these complexes can be internalized and favor macrophage infection, productive infection by macrophages. So this is seen in dengue. It's unlikely to happen with SARS-CoV-2 because macrophages cannot be productively infected by SARS-CoV-2. Another possibility of enhancement is enhancement of disease. And this would involve the activation of innate immunity, complement activation that would promote inflammatory cytokines. So the presence of antibodies would potentially exacerbate the inflammation itself. This is obviously not a black and white process. It's likely to be dependent on the balance between neutralization on one hand and the inflammatory properties of antibodies on the other hand. In this very elegant discussion in Nature, and Irving and colleagues discuss these possibilities, but actually make a strong statement about the fact that there is very little clinical evidence that disease enhancement happens in the presence of antibodies against SARS-CoV-2. So although it's something that we should bear in mind for future vaccine trials, for instance, the evidence today is not strong. And because antibody responses and the effector functions of antibodies are very complex, this complexity calls for broad, complete, multi-parametric assessment of antibody responses to SARS-CoV-2 in patients. And this is what these authors have done. They've looked extensively at antibodies and their effector functions in hospitalized patients. And what they found is that convalescent patients, so basically the patients who after hospitalization survive, have an antibody response that's biased against the spike protein, whereas the diseased patients had a response biased towards the nucleocapsid. So it seems to be important that, so first antibodies can be protective, we knew that, but it seems that a response biased towards the spike protein may be beneficial and prevent fatal outcome. So I will not dwell too much on T lymphocytes. This is what we saw in May. There was evidence that T lymphocytes in the context of severe infections are depleted and are dysfunctional with limited antiviral capacity. And actually this concept have been supported by the studies that have been done since. And if you are interested in those aspects, which are a bit complicated to discuss in a few minutes, I refer you to this very good review that was recently published in Nature Reviews. One important thing is that even if we understand better the pathogenesis of severe COVID-19, the relationship between the immune parameters and the risk factors that have been associated with fatal outcome remain incompletely understood. In this, I think, important study conducted in the Western Cape province in South Africa, the authors have looked at risk factor for COVID-19 death in a number of groups, either a large population or patients with COVID-19, whole COVID-19 cases or hospitalized cases. And what they found is like what was found in other countries: sex, age, diabetes, and other comorbidities were risk factors. What they found is that tuberculosis and HIV were also risk factor for fatal outcome of COVID-19. So this shows that depending on the population and the environment, different risk factors, in addition to the ones that have been well characterized, could be important, and that may involve immune parameters. The second point I wanted to discuss, and the two next points will be shorter, the second point I wanted to discuss is the role of exposure to non-SARS-CoV-2 coronaviruses, common endemic coronaviruses, and the induction of immunity to SARS-CoV-2 itself. A number of papers have shown that T-cell responses to SARS-CoV-2 peptides, SARS-CoV-2 antigens, can be detected in people who have not been exposed to the virus, either because they haven't had any sign of infection, or even people from whom samples were collected before the epidemic. And what these data, for instance, show that peptide pools or fragments of the N proteins can be recognized by cells from unexposed, uninfected people. So there is immunity to cross-immunity to SARS-CoV-2 in unexposed people. What we know since is that these responses are boosted by SARS-CoV-2 infection, and that the T cells express an effector phenotype, so they could have protective role. The possibility that antibodies could be induced in a similar way, cross-reactive antibodies could be induced as well, is very much debated in the literature. There is no consensus yet, but I thought it would be interesting to share this preprint with you. These authors have looked at convalescent subjects as compared to naive subjects, so serum collected before the epidemic. And so what they see is that in parallel to SARS-CoV-2 specific antibodies, they see that convalescent patients, so patients who have been infected with SARS-CoV-2, also have increased responses to other coronaviruses, suggesting that some cross-reactivity could occur at least in some patients. So we may not all be equal to SARS-CoV-2 because of our previous contacts with other coronaviruses. So that this is what these data suggest. Another parameter, immune parameter, pre-existing infection that might be important is the state of the innate immune system and what is called trained immunity, the capacity of the immune system to respond to a broad range of pathogens. And also, specifically, the role that vaccines like BCG could have by promoting this trained immunity and these innate immune responses. The possibility that BCG could have such effect is supported by a recent publication by the group of Netia and colleagues. And this is an interim analysis of a randomized controlled trial in elderly patients looking at the incidence of viral infections and respiratory infections over a period of 1 year. And so what this interim analysis is showing is that BCG vaccinated elderly patients have a twofold decreased risk of viral infections, especially respiratory tract infection. And the reason why this interim analysis was conducted is because there was an interest in testing BCG in trying to protect against COVID-19. So they had questions about the safety of the approach and potential efficacy. And what this study shows is that BCG is safe in that population and suggests that it has a protective effect. And we are looking forward to the results of those trials, looking at the impact of BCG on the susceptibility to SARS-CoV-2 and COVID-19. The third element of this talk relates to the immunity that's induced by SARS-CoV-2 infection itself. How durable it is a major question which relates to the occurrence of these recurrent infections that have been diagnosed over the last weeks and months. So what this paper shows is that actually IgG to SARS-CoV-2 in convalescent patients persists over a period of at least 3 months. So we now have more time that we can examine it. And so these IgG indeed persist at high levels over the following months, 3-4 months after infection, longer than IgA and IgM. I think it's important to emphasize that this study was involved primarily cases of hospitalized patients, and we know, and the literature is telling us more and more that patients with mild symptoms or asymptomatic patients may have a shorter duration of immunity. I will finish shortly. Just a few more points I want to emphasize about this emerging field, which actually has yet to emerge, which relates to mucosal immunity. And I think it's very interesting to see that in the 80s, chimp studies, so human challenge studies, were performed for coronaviruses, and these studies showed that IgA in nasal washing, as well as serum antibody, neutralizing antibody levels, were correlates of protection against coronaviruses. So we are looking, maybe in a few words, we are looking at the presence of IgA in the nasal wash of convalescent patients in a large cohort in Belgium, and we do see high levels of IgA in the nose of these patients, high levels in the saliva, and no big difference between the two sites, suggesting that saliva could be an interesting site for sample collection. We also see that T lymphocytes are induced by natural infection. So overall, we have immunity that's induced by natural infection, but the big question is how protective is this immunity? And so I will quickly mention that we are conducting a trial, a study at this time in Belgium, looking at 2,000 subjects that we will be following over the winter. Half of them will be convalescent patients, and half uninfected controls, to see whether natural immunity is protective, and because we look at baseline immunity to SARS-CoV-2 and other coronaviruses to identify correlates of immunity. Maybe one word about a study I like a lot that we are completing at this stage, and that's the effective transfer of SARS-CoV-2 antibodies to the newborn. We compare antibody levels in cord blood and maternal blood, and what we see is that those mothers who acquired SARS-CoV-2 infection very late during the last part of pregnancy are actually able to transfer SARS-CoV-2 immunity to their baby with antibodies promoting phagocytosis, complement activation, and NK cell activation. So I will be concluding here. So no, just emphasizing that moms have actually beaten big pharma because they've been immunizing their children already in May 2020, so well before pharma. So as a conclusion, I think we see a clear central role for inflammatory response, deleterious role, and defective entrant pathway in severe COVID-19. Neutralizing antibodies likely contribute to control of disease and are a good marker for protection in future vaccine trials. We are learning about the other functions of antibodies, and we still need to learn more. There is cross-reactive immunity with other coronaviruses, and this could contribute to SARS-CoV-2, need for more studies on mucosal immunity and for protective studies of infection. Productive studies of infection-induced immunity to guide vaccine development. So I think that to conclude, we've seen tremendous progress in our understanding and knowledge about SARS-CoV-2 infection and COVID-19, but you may feel that this progress actually doesn't lead to a major difference in the way we are dealing with this epidemic, because actually, like in 1918 with the flu pandemic, what is proposed is largely wearing masks and social distancing. I think that we will see in the coming months, we can be hopeful that the knowledge we have gained will allow us to develop new tools, therapeutics, vaccines that will allow us to deal with this epidemic in a very different way. I think a big challenge will be to make sure that these tools are accessible to everyone on the planet. And for sure, 2021 will be a public health odyssey. Thank you very much.
Thank you very much, Professor Marchand, for the extremely dense presentation, which we could fill a whole day, I suppose. But this is a question for later.